I don't know whether I should be spending another 10 minutes figuring out how to, well, share screen. There it is. So maybe this will work.
All right. Works.
All right. So, Mario, this is very much along the lines of the ideas I had shared with you at the All Hands meeting last November. But we've made some progress in moving in that direction, and I think we're at a point where we'd really appreciate some concrete help from you guys in a variety of ways. Also, that help could include saying that you've already done this, and so we should move on. Please feel free to interrupt at any point. I just put this together in 20 minutes last night, so apologies for the unpolished nature. I'll start with an overview of the basic idea of our center, which has really two types of data that are being analyzed. And our goal is to build a novel analytic framework that simultaneously looks at two types of data in making its analyses and predictions. The first one is what we call the knowledge network. I'll refer to this as KN. It's a heterogeneous graph where the nodes typically represent genes and proteins, and relationships among genes are encoded as edges. Also, the nodes in the graph include properties or annotations of proteins, and you can think of gene ontology terms and biological pathways from KEGG or other places, and the presence of a PFAM domain and so on. So biological and biochemical properties are also kept as nodes in this heterogeneous graph. And then you connect a gene to a gene node to a property node when there's prior annotation supporting that relationship. So that's a way for us to store a large body of prior literature as curated in various databases, literature and other data sources that have led to the creation of various databases. We create our own copy of an integrated version of these different data sources and store it all in a large heterogeneous graph. And this is going to serve as primarily as prior knowledge to inform our analyses of user data. So the actual thing being analyzed by our knowledge engine will be the user-provided spreadsheet of genomic data. Typically, you can think of transcriptomic data as a spreadsheet where the rows are genes and or transcripts, and the columns are various experimental conditions. I think in many cases, not just in transcriptomics, the user's data can results after some initial pre-processing results in such a spreadsheet being created, and the user wants to make sense of that spreadsheet. So that's shown in this cartoon also here. On the left is the knowledge network with its variety of nodes and edges in different colors. And on the right is the spreadsheet provided by the user for that particular session. And of course, there's a natural mapping between these two data types, which is provided by the fact that genes that appear as rows in the spreadsheet also appear as nodes in the network. And so ideally, the methods that analyze the spreadsheet should be able to make use of this additional information about the rows of the spreadsheet provided through the network. And I'll also mention that typically the spreadsheet is analyzed on its own using various machine learning methods, the data mining methods, and the network is analyzed on its own, typically using various graph mining methods. And there is scope for combining the two types of approaches into integrative methods that make better calls, better analyses of the spreadsheet. So we have, in the first year and a half of the center, we've made a very substantial progress in terms of collecting the data sources that go into the knowledge network. And in particular, there are the data from 6 well-characterized species, and edges represent protein-protein interactions, genetic interactions, co-expression, protein domains such as PFAM, gene ontology, biological pathways, TF binding, motif presence, homology, and so on. So I've listed some of the prominent current resources that we imported these kinds of data from in creating this knowledge network. I'll leave it at that, just to give you an idea of the kinds of annotations, prior annotations that go into the knowledge network. As part of the pilot project with the LINKS DCC, we last semester, Corey Post, who's sitting here, and I don't know if you can see, but Corey Post and Charles Blatty went about getting the LINKS data into our local databases, and develop the formats by which those data will be stored, and the tools by which those data will stay updated. So in particular, we focused on one aspect of the LINKS data, which is the transcriptomic signatures from the L1000 project. We downloaded these from the LINKS Cloud Amazon S3 service, and we used the LINKS API and software library provided by you guys in processing the resulting files. The data then got stored in the databases, both in the form of a regular MySQL database as well as a graph structure, where we added 1.3 million new nodes, each experiment being a new node in the knowledge network, and 722 million new edges, where the edges were the presence of a gene as a DEG, or differentially expressed gene, in a particular LINKS experiment. We so all of these, the database functionalities are containerized, and the tools for collecting them are all streamlined, so that at a push of a button we can update our local copy with the latest updates on your end. So this is just how the data is stored in the SQL form, but I'll move quickly beyond it, because most of our interest is in exploiting the graph-based representation of the LINKS data. So,
can I interrupt just with a quick question?
Yeah.
I wonder if you could elaborate on the assumptions or philosophy behind the choice of making each experiment a distinct node, because that's
when I propose the kinds of things we want to do, we want to enable, that will become clearer. At this point, that the choice is not well motivated. Except for the fact that these are different conceptual categories of information on the LINKS side. But you will shortly see in the next 3 or 4 slides that what we want to do with this kind of representation. Okay. And I can also mention that our primary goal was not to make fast queries to LINKS data available to users. You guys are doing a great job, and and have the headache of making that possible. Our hope is to tie the LINKS data with the particular paradigm of analysis we are building already, and ask if the kinds of queries that we are supporting in knowing can actually give us anything new or interesting on the links data. Does that make sense?
Yes. Thank you.
So, as I briefly mentioned, we represent each links experiment as a node in the knowledge network. Actually, there are 3 kinds of nodes that are created from each experiment. There's one node called the links experiment node, or LE node. So there will be 1.3 million of those. We already have gene nodes in the knowledge network, so we don't create new nodes for those. We just link or connect the links experiment node to a gene node if the gene was significantly up or down expressed in that experiment. And thirdly, we have these links condition descriptor nodes, or meta information nodes, such as the discrete information about the perturbagen, cell type, perturbation time, and dosage, etc. So each links node is this is a cartoon of that. In the middle is one links experiment expressed or represented as a single node, and it connects to a variety of. On the left, it connects to the LCD or links condition descriptor nodes, and the right, it connects to the gene nodes, which are already there in the knowledge network. And just a technical detail, for now, we have connected a gene to be to a links experiment node if the z-score of its expression in that experiment is greater than 2.5 in absolute value. And this may not be. I don't know if Corey followed guidelines from your end, or if whether you guys are imposing any such thresholds anywhere. Maybe we just did it to get started. And if you guys tell us that this is not the right thing to do, we're happy to. We would like to update this. But for now, I think we need to represent the information in a links experiment, the DEG information, as edges, and so we have to threshold. We do retain the weight, the z-score of that gene as a weight on the edge, so that an algorithm that's capable of exploiting edge weights can utilize that. All right. So what are the kinds of things we are trying with this once this sort of representation has been achieved, which it has, and so we are on to these projects in the early stages now. First project is to make a gene signature network informed. So this is a very simple idea borrowed from recent literature on network smoothing and its application to, let's say, subtyping of patients. So what we do here is you have a gene signature as in the form of a set of genes. You perform a random walk with restarts, or RWR, on the knowledge network. Again, the knowledge network will include all these property nodes that represent gene annotations. You do a random walk with restart using these DEGs from that signature as a restart set. To illustrate this, so you have the knowledge network. I've drawn this as a homogeneous network, but in reality it's heterogeneous, and we've been worrying about what to do with heterogeneous graphs. The user specifies the gene set. In our case, the links experiment, the query gene signature amounts to a gene set, the differentially expressed genes. And then you do an RWR on the network, and with this as the restart set, and this will hopefully result in related genes being identified, or guilt by association, where relatedness is as defined by proximity on the network. And so once you have this, and to be a little more technically detailed, what we gather after the random walk is the equilibrium probability distribution on all nodes. And so once you have that you can think of that as a smoothed version of the gene signature. and that has incorporated prior knowledge about gene-gene relationships and gene annotations. We've implemented this network smoothing, and the next step is to, so the goal was to ask if the task of finding the most similar signature for a given user query signature, can that be improved upon by network smoothing? And so we are on to running these. We're not going to do this on all 1.5 million links experiments as our signatures. We're going to take some small subset to evaluate the method on, and then try to show that smooth signatures group better or cluster better, perhaps, lead you to better query similarity results than unsmooth signatures. This is a hypothesis that it's not. It has to be really tested. It could go either way, and at the moment we are struggling with defining how to do a benchmarking for this. And so this is the first question for you guys, because you have, on your side, you've published variously on similarity metrics and query responses with signatures. How do you demonstrate the accuracy or estimate the accuracy of these kinds of signatures within your framework?
So can I just ponder something there aloud? So presumably your network encodes all the information we have about these entities, right? In this way, it's going to encode basically relationships between different signatures, because they're going to be connected through the commonly differentially expressed gene, right? So,
right.
So at the end of the story, go ahead.
That's a good point. In this particular project, this network smoothing is not using the rest of the links network. It's only using the when you do this random walk, you do it only on the prior annotation knowledge network. You keep the links data out of the random walk, the links nodes out of the random walk, except to define the starting point.
Is that for this particular project?
For this particular first project. We will, in the next project, bring in the links nodes in a full-fledged manner into the random walk. So, Mario, does that change your question?
Yeah, I think it does change, but I guess not. I'm not sure how much it changes. So if you remove all the nodes that the node experiments, and then these nodes are actually connected to these property nodes, right? That define the type of perturbations and all this kind of stuff. So basically, effectively, none of these information will be used then in smoothing. All right, I understand. So the only information that's going to be used is defined by your general knowledge about the gene, right? Is that
right? That's right. In this case, the way links data is being used is take a user gene signature and smooth it on non-links data, using the other annotations. Take a linked signature and smooth it on non-links data, again, using annotations. And then compare the two signatures for a similarity score, thus deciding a similarity metric between each link signature and the user's query.
All right.
So the right term to call this might be knowledge smoothing, or maybe knowledge regularized signatures.
That's a nice word. Right. So that's the idea. And I think you understand then that we are in this predicament of how do we demonstrate an increased value without having gold standards or things like that. One thought we've had is that a similarity score should track with the links experimental annotation. So if we use a links signature as a query, then it should end up being similar to, our similarity score should place it as being similar to something with the same metadata or perturbagen information. And so and we can do this on a large scale to ask if links experiments of similar metadata end up showing strong similarity to each other. And I've asked my students to read the papers from your side in which you propose similarity scores, which I know, and how you evaluated them, which I haven't followed yet. So, but I know that you must be doing this already. So any advice here would be helpful.
Well, I guess to me the first question, because this sounds all very interesting, but the first question would be, what is the correlation of signatures before and after smoothing or regularization?
Okay. Yeah, that's a valid question, and so if it tends to be no different before and after, that's something learned, and if it's very different before and after, then we have to ask which one is more informative or useful. And so yeah, that can be done without any benchmark, and we can report on that as soon as we have some numbers. But how do you then go beyond that, assuming that there are differences and it's not perfect correlation? How do you go? What kind of? So, for instance, if you take two links experiments that are very similarly annotated, so slightly different time points and everything else being same, the fact that you've placed them at a high similarity may not be so interesting, right? So what makes an interesting benchmark of links experiment similarities to work with?
Right. So now, if you're working with KD, is that what you said that you were doing? Oh no, this was called you just differentially expressed. I mean, there are some natural things that you could do, right? Like if you're looking at knockdown signatures, you could look at whether the ones that are in the same pathway are closer, right, than before. If you're looking at drug signatures, there are various categorizations of these drugs that would define sets of more similar and less similar drugs, right?
Yes, you can classify them based on, or cluster them based on their chemical similarity, and perform.
Resource such information. We are relatively new to this area and have found multiple resources about annotating drugs and grouping them. So where do you go for drug grouping?
Oh, you can start with something simple like ChemSpider.
ChemSpider.
Mm-hmm. It's available online, so you can. There are all kinds of measures. You can find a software if you want to run something locally. There's a software package called Babel, just like the Tower of Babel, that has Tanimoto and some other coefficients, similarity coefficients that can be computed for a pair of chemical moieties. Okay. So there's a couple. I can send you some links later as well.
Okay, I can't see very well. Is that Jarek?
Yes. Yeah, I'm here.
Yeah. Great. This is very helpful. And are you guys have you guys explored anything similar in the past?
Yeah, I can tell you from my attempts using different things that actually self-recognition. It sounds trivial, but it's always something that is worth looking at. Meaning, if you can really discern your the signature that you're looking for against other signatures, and what is the degree of success in this experiment.
That's good to know. And even the ability to highly score yourself or something highly close.
Yes, and measure the difference between your own score versus everything else, right? The distribution behind. When you treat all other signatures as unrelated, although that's not entirely true, there's going to be a tail of the distribution, but nevertheless, score for your own signature, query signature versus mean score for all other signatures.
Yeah, very good, very good. Okay. Okay.
So can I ask you just for a couple of clarifications in terms of how do you actually define your signature? So you are saying you take the genes that are differentially expressed, right, in each experiment.
Yeah.
And these sets now define your signature.
Yes, that's right. One set is one signature.
Okay, so you have one signature is the set of differentially expressed genes.
That's right.
And you plug them into your network.
With Z-scores, right?
Right. So that depends. Yes, the Z-scores will go into the edges in the node, but
and then they find
they are already there. How do I say this?
So the Z-scores could be, in
No,
the picture that he's showing, there's an equal probability of starting at any of those.
Right, they're not. Right.
The Z-scores could be used to make a non-uniform probability where very strong differentially expressed genes are the most likely to start.
Sorry, yeah, I misspoke. So there's two ways. The simpler way is to think of the gene set as a signature. That's it. And when you do a random walk with restarts, what it means is that anytime you decide to restart the random walk, you'll pick one of these genes in the set with equal probability to restart from. If you keep the Z-scores along with the genes, you have a gene set and there's Z-scores, that's another signature, kind of signature, in which case the random walk will restart with at one of these nodes with probability proportional to that Z-score or something related to the Z-score. So that's how you handle different levels of differential expression if you want to. Although we haven't really demonstrated any, we haven't really gone on to the more nuanced, non-uniform scenario.
Okay, so you have now. So your signature basically defines the frequency of restarts, and your algorithm always starts with your random walk. Always starts with one of the nodes that is one of the genes, right? So are you using 20,000 genes, or you're using just measured genes?
So we had initially started with the plan of using the 20,000, which means using the restarts from your imputed values across those 20,000. It's like whether, we would want the random, I think we would want the walk to be on the 20,000 because the network captures all the known relationships between all the known genes.
Yeah.
But if you think that's one of the open questions is, should we have focused on just the 1,000 measured from each experiment instead of the 20,000?
We'd ideally like to do the random walk on the 20,000 genes, with the restart set being a subset of the 20,000 and not just the 1,000.
Okay, so I'm just trying to get the basics. That's kind of already a fine point, right? But so you have, say you just have, you can start from any of them, and so you're. All right, so you defined the start, your differential expression defines how likely you are to start your random walk from any given gene, and your asymptotic probabilities of being at this node, right, define the new signature.
Yes. Yes.
All right. So the new signature is basically set of 20,000 probabilities, right, that sum to 1.
That's right.
Okay.
Yeah.
And then your similarity measure that is going to have to put these genes together and will have to operate on that vector, right?
That's right. We haven't even decided on that similarity measure. It could be the same similarity measure that you guys, the kinds of measures you've been using, so that one aspect is controlled. The only difference is in the smoothing versus not.
Okay. I gotcha. And you're using individual, so you're using 1.3 million vectors. So basically, you use individual replicates, experimental replicates. You don't actually combine in any way the replicates, right?
We need help with that. In our dealings so far with the linked data, we've been a bit lost as to what to in combining multiple replicates, or even combining or extracting from aggregating multiple time points and so on. So is there something intelligent we could do that avoids having each experiment as a replicate? And
well, I mean, so I'm just kind of trying to figure out, so because if you're using, in a first kind of approximation, right, if you're using each experiment, your first test would be, well, can I put together, replicates of the same experiment, right? That's an obvious one.
Okay.
Right, because each, then I would probably, so Broad Institute has created subsets of these signatures that they think work, right? They call them gold signatures. I don't know if you guys looked at, there is a metadata field that says whether its signature is gold or not.
Okay. And what does that mean? Sorry, we're
well,
not.
it means that they think it worked. They got good signature.
Which involves comparison of replicates.
Right, which involves comparison of replicates and some QC measures and that kind of stuff. So maybe it would be, I think there is merit in looking at everything, right? Because, for example, you may be able to make connections between lower quality signatures, right? That they cannot because, you know what I mean? Signature may be noisy for their methods, but it may work better in your method.
Exactly. Maybe you will be able to identify replicates that are not within the gold subset.
Okay.
With some reasonable accuracy. Yeah.
Okay.
Right.
Comparable to that for the gold subset.
It's a sort of a consistent signature. Okay, that's what you mean by worked. Yeah.
Right, and then so that could be the first pass, kind of benchmark. If you just try to put the replicates together, then if you ask yourself, well, what is the best way to actually construct a signature for a specific drug for a specific time point cell line, right? That we say, I mean, the way we are doing it at the moment is that we actually create like what we call level 5 signature, right? It's not something that they release, but we aggregate replicates of the identical identical experimental conditions and come up with a kind of super signature, right? Or we call it just a signature, right? And but that's not clear. Broad is very kind of cryptic about what they really think should be done. And but in my mind, if you're trying to now go from benchmarking based on just on technical replication, right, to benchmarking that's based on, well, are signatures of similar drugs more similar, right?
Yeah.
I would guess my guess is that it would be better to aggregate these replicates into signatures and then operate on these aggregated signatures.
How do we? Have you done this? And we are not aware of level 5.
Yeah, it's not, that doesn't exist, but it's kind of conceptual level, right? But we have these, yes. We have level 5 signatures. We created kind of what we think are good aggregated signatures. So if you would want to, say, try experimenting on these, we can give you.
Oh, that'd be so good. Yeah, absolutely.
Yeah.
When you query, querying.
Yeah.
That level,
right.
What's the question?
No, we were just chatting here that when you're actually querying iLinks, the signatures that you see are those aggregated ones. So you at least for some experiments, you could use just iLinks to see what the differences are.
Right, but yeah, I mean, he doesn't want to query one by one, right?
No, but just for some experiments.
Yeah. And we are in the process of updating this database because these are like 3 years old and so on. So there is a special version.
So when you update it, it won't be a dramatic change in the formats and stuff, right?
For iLinks?
Which database were you talking about updating?
I can't really hear you, Saurabh. I'm sorry. Can you repeat?
I heard you were talking about updating the database, and you haven't updated in 3 years. Which database were you talking about?
Oh, I'm sorry. It's not 3 years. It's like maybe 2 years. 2 years. It's the database behind iLinks, right?
Okay.
So we have, in the meantime, we've done a lot of processing, benchmarking, massaging, and now we are ready to kind of refresh our backend databases. But I don't think that affects you much because you probably don't go and point and click through our portal, right? So
no, not in the stuff you're hearing about. Some of our students do use the interfaces directly, where they want to check something. But here we've downloaded it from, this is not point and click. It's
right. It's a batch thing. Right? Yeah. So we can give you, the big tables, of things that we created that are gonna eventually or very soon be also updated on the iLinks, but they are similar, right? But it's more up to date.
They're aggregated versions, aggregated across replicates.
Right.
That'd be very useful for us for the second level of benchmarking. I understand that the first level will look at replicates.
Okay.
Okay. All right. Very nice. So the second project that we have not yet made concrete progress on actually involves random walk on the links graph. So I showed you this part in a slide earlier where you had the links experiment nodes and the links that's in the middle of this shaded area, the condition descriptor or meta nodes and the gene nodes on the left and right respectively. And then this is the knowledge network where you have the gene nodes connected to various annotations. The idea is to do random walk on this integrated graph to identify relationships. And so what kind of relationships? Well, one example, each of these 3 bullets is an example of a query. So you start with the links experiment node and using that as the query, you do a random walk on the integrated graph to return other most similar nodes in the KN, in the knowledge network. So you will go, I don't know if you can see the mouse pointer, you start from here, that'll be the query, and then the random walk will go through this layer of genes, and it'll even go back to this layer of links experiments, go back to the genes, and it will ultimately make its way to these annotations. And an annotation that's visited, with higher frequency in the asymptotic sense is deemed as more similar. So so it's the usual PageRank kind of philosophy of returning relationships based on random walks. In this case, we are trying to connect a links experiment to specific biological properties that are, such as pathways and geoterms that are heavily involved or significantly involved in that experiment's differential expression. So that's one example. The second example is you start with a gene set and you find links perturbogens. So wait a minute. So you start with a gene signature as a query and learn the most associated conditions. So you start with a subset of genes here and then go through the layer of links experiments here to specific links descriptors here. So for instance, the user's query set of genes seemed to be going up or down in experiments that involved knockdown of genes from knockdown of genes in this cell line or something like that. So a links condition descriptor, it could be a drug, for instance, that is found to connect to multiple genes here through multiple experiments. So it's kind of aggregating multiple links experiments in taking you from a gene set to a perturbogen. And the third example is you start with the links perturbogen and go all the way across two intervening layers. You start with the perturbogen and you go through all the links experiment and all the gene nodes connected to those links experiments to annotations that are connected to those gene nodes. So it's kind of two layers of indirection. This perturbogen results in differential expression of genes that are annotated with a specific term. So these are the kinds of connections we'd like to make using a random walk on this integrated graph. And the technical problem is that it's a very large graph. And so for this, we had to implement random walk with restarts, in fact, a PageRank algorithm for distributed computing on the cloud. And so it's been done, but there are some fixes that need to be made, and which will be done hopefully in a week or two. And so in March we hope to start doing some of these tests or some of these runs of on the integrated graph. And again, the question is, what are the most useful queries other than similarity of gene signatures? So here you can see that we are expanding beyond just asking for similarity of gene signatures to connecting one concept to another concept, a perturbogen to a geoterm via all the links experiments and all the gene nodes. So we have to obviously construct a suitable benchmark a priori, or at least a way to benchmark, and that's something that we haven't sorted out. Any comments on that project?
Well, I mean, it seems reasonable and kind of logical. I think benchmarks, to come up with the good benchmarks will be challenging because now you're kind of in a hearsay world, right?
Yeah.
A lot of the benchmarks that are being used are kind of based on, some. Well, anyway, but I think it's something that it's worth thinking about, right?
Well, I have to tell you that I like this a lot, but again, in terms of simple first steps in terms of evaluating the results, I would consider, especially for gene set enrichment analysis. So asking a simple question, to what degree are the gene set enrichment analysis concordant with random walk-based sort of annotation set, right?
Yeah. No, we've been doing that in other contexts. So you can think of a gene set enrichment analysis as being fairly closely related to, think of just the rightmost two layers. You have genes and you have an annotation. And a gene set enrichment analysis asks, you take annotation 1 and you count the number of edges that connect to genes in your set and ask if that's significant. That's what gene set enrichment analysis, typically is, for instance, through hypergeometric tests, right?
Right, yeah.
So now, and so if you do a random walk with that set of the restart set, you will have a high probability of ending in this node. And what our random walk is going to allow you to do is to extend it to testing with multiple annotations at the same time, so that and also extend it to having a multi-layer thing. And the direct comparison we've already been doing, in context outside of links, where we've used hypergeometric tests for in a number of papers in the last 10 or 15 years, and now we are asking, to what extent the random walks also reveal similar insights. And I think that certainly should be done again when we work with this with the links graph. But I think there's opportunity here to go to places where gene set enrichment cannot be performed because there's multiple types of entities involved. And not just the gene set and the annotation, but at least 4 types of entities: the perturbagens, the links experiments, the genes, and the annotations.
I understand, and actually this is why I like it a lot. But it would still seem to me that comparison wherever you can do this with gene set enrichment analysis to show that you get similar results, and then say, and by the way, we can do this also when gene set enrichment analysis cannot be applied. That should be probably right.
No, I'm totally with you, Jarek. I like the gene set enrichment approach because it's a very interpretable thing that is that can help in a large number of situations. And anytime you do something multi-level like this, you have to argue that this is worth doing beyond what the simpler approach would have given you. And I have no, again, I'm going into this with an open mind. Maybe all this will just not be manageable and will not give us the right signals. And just we'll find out. I think that's the nature of the study.
So Saurabh, if kind of just being philosophical or cynical about this, it's a nice idea. You may spend many years trying to rigorously prove that it works, or you can just create a nice point-and-click tool that people can do this analysis and buy into your logic that this makes sense, perform, and become famous. So you know what I mean? It's proving that it's really good and it gives you something on top of, say, simpler analysis. Right. Will be challenging. I think, at least in my experience, these kind of things are always challenging to prove that you are really coming up with something meaningful. But if you let people, kind of use your tool just by telling, look, this is logical. This lets you do things that you cannot do otherwise, and this gives you something, right? And you then go and see whether this something makes sense.
That's a great viewpoint, and I'm happy to buy into it right away. So, which means for us the priority is first construct the conceptual framework, which is sort of what is here. Second, provide the tool. And third, try to write a paper, if possible, that applies it and shows something interesting. For that third aspect, we'll need to have some idea of not, I shouldn't call it benchmarking, of some idea of evaluation. And I think what's nice is in this scenario is that there are studies that link a perturbogen, something on the left end of this graph, to a pathway, something on the right end. And it really doesn't map into a gene set enrichment question because it's not clear what the gene set ought to be. The perturbogen, according to links, has been examined in 200 different experiments, not one experiment. So it's not obvious what the unique gene set should be. The biological pathway, on the other hand, has been annotated with a certain set of genes. So it's not clear. There's some level of aggregation that's required anyways if you do a gene set enrichment approach. And in this case, because people studying the drug response are interested in the pathways involved, there is literature evidence that will tell us whether we found something, anecdotally at least, that makes sense. So I think it's valuable here, to ask if we are able to connect something on one end of this graph to something on the other end correctly.
Absolutely, I agree 100%, and I think I think there are. You're right, and that there are good actually gold standards that you can, or vague at least gold standards to some extent, that you can apply there, and it should be doable.
There's something called, sorry to interrupt, but there's something called BindDB. I don't know if you are aware of that. This BindDB database has a bunch of experimentally validated binding affinities for drug-like compounds and their biological targets. And you can actually use that maybe as a bigger set of compounds for which the actual mode of action is hence defined.
Oh, great. Okay.
And then you can use those to
connect pathways as well. All right. Yeah. No, I think Casey, for instance, you were looking, what database do you go for pathways associated with drugs or genes associated?
GKB is
Farm GKB is one thing you.
Okay. Yeah, that's also reasonable.
Yeah. So exactly, there are annotations that we could try to recover, by using this. I mean, we are short of time. These were two of the main ideas. A third idea is, let's see. So, is based on enrichment tests entirely. So the idea here is take a gene signature and test it against for enrichment against various biological processes and properties. And this is the kind of thing that enricher would do, for instance. And we can use enricher, or we can use an in-house version or a copy of enrichment test tool. But then that gene signature is we represent it as a bag of enrichment. So it's a set of terms or properties that were enriched in this signature. And then it's like a representation of the signature in a new space. And we start trying to see what this space is giving us. It's kind of a dimensionality reduction or dimensionality transformation going on. For instance, you can try to cluster signatures in this new representation space that was based entirely on hypergeometric tests against biological processes. And then, this is sort of related to the first project. Do we find expected or unexpected groupings of signatures when we look at them not at the gene level, but at this at this enrichment set level? Does that make sense?
Yeah.
So that's the project that Jinpeng here is working on, and it's there's no random walks involved here. In principle, you can replace the enrichment portion with other approaches, including random walk approaches. For now, he's just working with hypergeometric tests. The main point being to figure out how to examine the value of this new representation of a signature. Yeah, go ahead. I'm done.
I was just going to say we will have to run because we have another meeting at 3 that's supposed to start. So we can be late a couple of minutes, but Avi, Avi, right? Avi Mayan, he's been actually working on on these kind of things as well. I mean, you know, historically many people have been working on that, but I think he has something actively working on, and you know, it's like maybe conceptually similar, but the details are different. So, and maybe he has a paper. Does he have a paper on that, or?
Either already published or in the
right.
pipeline. Yeah.
So it would be worthwhile kind of seeing what he's done, maybe as a starting point for you guys to conceptualize where you're going.
Okay. No, we do follow Avi's publications to the extent we can. As long as they're public, you know. But I'll, you know, if he's doing anything new that's not published, you know, maybe you guys can point us to that. We certainly don't want to repeat things.
Well, I mean, look, it's kind of, I think it's something that you would want to do in your hands anyway, with the idea that you're going to expand this to maybe new profiles, right, that are based on network smoothing or whatever later, right? That is not just enrichment analysis based. So I think it makes sense for you to do it as well. It's just I wanted to point you that maybe he has something out that is really worthwhile looking at. That's all. You know? And if I'll take a look, and I'm not sure if the paper is out or he has some kind of tool, and we'll send you a link if you find something.
Thanks, guys. I know you have to run. I'll write an email shortly to summarize and plan the next steps. And one thing I'd hope is, I mean, I've explained many ways in which you guys can help us and have helped us. If you can find ways in which we can help you, please let us know. And that can be over email.
Okay, sounds good. Maybe we'll focus on that now, how, you know, but this is a great beginning. I mean, it's not just beginning, but I think we kind of already see some.
Okay.
I think those knowledge regularized signatures, that's really a great way of making this next step and maybe making them available through iLinks or.
Thanks for the cool name.
Okay, guys. Well, thank you very much. That was a lot of fun, and we'll talk more, right?
Yeah. Bye.
Okay. Bye.
Bye. See you guys.